So maybe I will wait for a couple more minutes because the turnout wasn't very huge last time also. So we wouldn't waste much time and we'll just begin with what I have prepared for today. So just a couple of more minutes we can wait and we can see whether or not anybody's joining or else we can start and the recording is going to be available later.
Thank you, madam.
So I hope your session with Sir yesterday was nice. I was unable to attend that particular session, but hopefully it was nice and hopefully there were a lot of doubts that could be solved over there.
Most of the discussion was not around the doubts in the course. It was around functional genomics and things like that. And definitely, he was able to clarify. And this is the first time I am seeing, instructor, NPTEL instructor, giving an opportunity to meet, as I mentioned even in the last session. That was very nice of him. And definitely, there are plenty of questions to be answered.
So there's always a time constraint that is there when Sir is taking a lecture. But so he makes it a point to take, like he has all 4-week courses. So at least 2 sessions per courses every year. Even in the reruns, he tries to take. So that is always one positive when you are attending one of his courses.
That was very nice of him and nice of this course itself. I'm taking other 4 courses. Only one more course instructor comes. Others, they don't have even problem solving session also. So we are left just like that. So compared to those things, you people are doing great. That's what I wanted to say.
This part actually has been the credit for the problem solving session, not by Sir, but by the TAs. That side, it completely goes to NPTEL because this is something that they have coordinated. And the instructors, the time that they take out, that is a collaborative effort of both NPTEL and them. So, okay, a few people have joined. Maybe we can just begin with the class. If there would be more people joining, I will keep admitting them during the lecture itself. Just checking. Yeah, so the recording is already on. So I record these lectures because these are required by NPTEL and later they can hand over these recordings and the material that I will be discussing, I will hand it over to the to NPTEL. They will later maybe give you a link to access the lectures, both the lectures material wise and video recording wise. So let me begin with a brief introduction of myself. Okay, first I'll start sharing the screen and first I'll introduce myself because I see a few new names that people who were not there in the previous class, right? So first maybe I'll introduce myself a bit. All right. So hello, everybody. I am one of your teaching assistants, Arveen Kaur. So I'm a TA for this course that is Functional Genomics that is being taken by Professor S. Ganesh from IIT Kanpur. I am also a PhD student myself at IIT Kanpur. And coincidentally enough, I am one of Professor Ganesh's students. And apart from this, let me talk about how basically I have structured these problem solving sessions on my end. So what I do is that I have broken it down into two presentations, which I estimate will take 1 hour each, plus minus 10-15 minutes, right? So in the first presentation, what I do is I take up the assignment for the last year. So right now you have an ongoing pending assignment for week 2, right? Just a moment, let me add whoever's trying to join. Yeah. So right now you have a pending assignment for week 2, right? If I'm not wrong, it has to be submitted in this week. I am not sure about what particular date it is. Right. So I cannot really discuss that particular assignment with you because I can't give out answers. But I can discuss the kind of questions that were asked from this week's coursework last year. And there are some recurring questions also from time to time. So it might directly give you some answers at least. Also, along with discussing these questions, we will go like if there is any concept that has to be clarified. So we'll go and delve into that concept also briefly where it is required, right? So that will be the first part of my discussion today. And the second will be uh, extra concepts and questions, right? Or some doubts that the people put in the discussion forum that I usually discuss. But I did look at the discussion forum this time, and most of the doubts were pertaining to that of the first week's assignment itself, and that had been clarified by the TA, right? So, and that had already been taken up by me last time also. So just a moment, let me stop sharing so that I can join whoever is trying to join. Yeah, so let me stop sharing. Okay. Yeah. So since this week there were not many questions from week two's lectures, I have myself picked out some topics that I thought would require a little more clarity and it would help you maybe in the long run. So we'll discuss them in the second part of the presentation. Also, a few disclaimers that I would like to give prior to beginning with my presentations is that one, since we are meeting on Teams, right? Just a moment. I want to check if, yeah. So since we are meeting on Teams, which is one of the platforms had a premium version of, but there is a restriction over here that since I have not added you people by your email IDs, the chat box will remain blocked, right? It will not be accessible to you guys. Even if I write something or you write something, I will not be able to see that. Okay. So let me just admit these people also. And I reshare. Okay. So since the chat box is inactive, I would request one thing of you guys. That is, if at all there is a doubt between the presentation, right? Maybe you can interrupt and maybe I can try to solve it right away. Also, since I am not an expert in functional genomics, I am still studying and thriving to be better at it. I might not know the answers to all of your questions. So the things that I will not be able to answer today, I will make it a point to go back, check it, and maybe I'll get back to you in the coming week. So that was it for the introduction part from my side. So I will begin with my presentation. Yeah. So also, if at any point my slides freeze, because that sometimes happens because of internet connectivity or when I'm admitting people, which I think I have to do right now. Yeah. So although I always make it a point.
Admitting of the people can be done by anybody, madam.
Are you getting that option of admitting people?
Yes, madam.
I'm not
So
sure.
you please continue. You continue. We will admit the whoever comes in
Okay,
the
okay.
lobby.
Okay, then that is fine by me. And I think it will be much less interruption also in that case. Um, okay. All right. So I hope my presentation is visible to all. And so first we are going to start with the previous year's assignment for week 2 for the course Functional Genomics. And we have 10 questions in this assignment. So how I've prepared the slides is that first I will just put
Your
forward the
screen is not
question.
visible, madam.
Oh, it's not
Screen is
visible. Okay. I'll reshare.
We can see you. We can see the screen as well as you.
Okay. Nonetheless, let me just reshare.
now okay.
It's okay. Okay. Yeah. So I will read out the question, read out the options. I will give a moment to you guys to think about it. Since we do not have an active chat box, we cannot go for about it as a quiz, right? It would have been much more interactive that way. But then we'll basically come to the answer and discuss the concept behind it. So the first question in last year's assignment was, in a polymerase chain reaction, that is a PCR, how many DNA duplexes can be obtained at the end of 4 cycles if one uses 2 DNA duplex molecules as the template DNA? So I hope everybody is aware of what PCR is and how we will basically count how much uh, outcome we are getting after a particular number of cycles is what this question is about. So your options are whether it will be 8 or 32 or 16 or 64. So think about
B,
it since
B.
B. Okay. So, okay. Yeah. So the answer is B. How it will be B, right? So if at all anybody does not know this concept, we will look at it both visually and with the formula. So visually, if we have to see, what will happen is that there will be a template DNA, right? There will be a set of DNA that will be present. Just a moment. Okay, I will just use my pointer. So there will be a set of DNA that will already be present. Okay, this is the DNA duplex that we are already talking about. Right, so this green represents the DNA, and over here in the first cycle, this red represents the primer getting extended, right, and forming an amplified version of DNA. So after the first cycle, how many copies each? Right? So at the end of the first cycle, we will have, instead of 1, we will have 2 copies of DNA. Similarly, at the end of 2 cycles, instead of 2 template strands, we will now have 4 template strands. Like this one is 1 template, this is another template, this is the 3rd template, and this is the 4th template, right? Each template will contribute to 1 other copy, right? So one more complete DNA will be able to form. So it will again double and form 4 copies. Similarly, in every cycle, there will be a doubling that will occur, right? So at the end of 4 cycles, for instance, since we have 16 templates, we will at the end of it have 16 copies. Right. So what is the formula of this particular reaction? It is 2 raised to power, just this 2 raised to power number of the cycle, right? So number of cycles is 4. 2 raised to power 4 is equals to 16. So we have 16 copies. Now this is when we had 1 DNA template or 1 DNA duplex to begin with. Once we have 2 DNA duplexes to begin with, so we can just double this number and the formula will change to be 2 into 2 raised to power number of the cycle. So instead of 16 copies, we will now have double that, which is 32. So I hope it is clear. Also, okay, just because I have not put in that illustration and just for revision purpose, I'm almost sure that everybody will know this. But what are the 3 steps of DNA? Okay, so I think somebody is trying to join. Can they be admitted by anybody else? Just checking. Just wait a minute. I'll do that. Anyway. Okay.
I admit you, madam.
Okay. Thanks. So what are the 3 major steps in DNA? Like, majorly speaking, what are the 3 steps that occur in a PCR cycle? Okay. So the first step is separation of these two, right? It is denaturation or separation of the two templates which are forming a duplex, right? The second one will be annealing or going and binding of the primer to a template. And the third will be extension. Okay, extension will mean that after the primer is bound, the dinucleotide phosphates, they can come and they can keep binding by the action of DNA polymerase enzyme, right? Stack pol over here. So the 3 steps are first denaturation, then annealing, then the third one is extension. So moving on to the next question, it is which one of the options is not true with regard to morpholinos? Okay, so another disclaimer that I would like to give is that these questions have not been framed by me. So there, like in this particular question, there is some kind of a mistake in the options, mistake grammatically, right? But since this was what was presented last year and there still can be a valid answer to this, so maybe just neglect that and find which is the most suitable option to what is not true with regard to morpholinos. So the first option is a phosphorodiamidate morpholino oligomer. Yeah, what happened? Sure, yeah. Is my screen visible? I don't think so. Wait. All right. So we were on question number 2, and the options for which one is not true with regard to morpholinos was: is it a phosphorodiamidate morpholino oligomer, or can it be used for gene silencing applications, or can it block the translation, or can it integrate into the genome? So which one regarding morpholinos is not true? So I think it is a very obvious answer. If you had gone through Sir's lectures nicely, you would already know that morpholinos are basically modified oligomeric molecules. They have a different backbone than that of DNA. And one thing that they cannot do is that they cannot integrate into the genome itself. They can be used for gene silencing. But what does gene silencing basically mean? Gene silencing means that whatever activity suppression is happening, it is happening at the level of either the mRNA or the protein translation, right? It is not affecting the genome itself. Okay. So morpholinos are small single-stranded oligonucleotide sequences, and they do not get integrated into the genome. So if we look at a morpholino, this has some major changes than a normal phosphodiester DNA. One major change is that instead of this 5 carbon ring, it has a 6 carbon ring, right? Also, instead of the
Madam, they are
normal
synthetic or natural.
what? Can
Morpholinos
you
are
please repeat?
synthetic. That is a big difference between morpholinos and that's why they cannot integrate in the genome. They are
Yeah,
synthetic
yeah,
or
yeah.
natural.
They are synthetic. So this has been created synthetically so that they can interfere with the mRNAs or the protein uh, translating machinery, but they cannot basically integrate themselves into the genome. They have been synthesized like that. And usually they are around 24, 25 units in length, right? So, for example, if we just talk about another kind, for example, RNAi, right? Any such peptide or any such substance that is used as an RNA interfering molecule is usually around 12 to 13 subunits or 12 to 14 subunits, while morpholino is around 25 subunits long. Right. So what happens? What this is one of the advantages of using a morpholino, because what happens is that it will be very, very specific. Right. If you have more number of subunits, means that there is a particular sequence that it can recognize. Right. If the chances of a smaller sequence getting repeated by chance in a genome is more likely than a large sequence getting repeated in a genome, right? So if a large sequence, if there's a this peptide that is formed to basically go and attack to a large sequence, it will basically be very, very sequence specific. It will not have any side effects, right? If we look at roughly what morpholino does, so it is basically again this kind of oligonucleotide. This is a general DNA that is represented. It is not a morpholino. Morpholino is represented over here in orange, right? So over here, what do you mean to show is that from the DNA, mRNA was formed, right? This RNA could have been matured to make an mRNA. Now, this mRNA is present in inside the nucleus, and if it is mature, it has come out to the cytosol, and there they can come and interact like this morpholino that have synthetically been made and transferred into the cell can basically interact with a sequence homologous mRNA, and when they interact with that, another they will basically block the either the reading of that mRNA and its translation into the protein, or there could be another pathway that has not been depicted over here, that they can directly hamper the protein making machinery also. Like they cannot come and sit on the mRNA, right? One, it might be possible that the morpholino is present. That is why just that particular, like maybe from where the reading has to start, that start side is hidden. So the translation cannot happen because that signal is not being transmitted. Secondly, what can happen is that maybe that signal for translation has been transmitted, but the morpholino has come and set in such a way that the machinery that is required for basically reading that sequence cannot come and sit on the mRNA itself. So the amino acids can also not be like the codons cannot be read again. So the third question is, which one of the following options is true with regard to the step that is highlighted as a red rectangle in the given schematic? So this red rectangle is what we are talking about. So is it degradation of peptide being made, or degradation of the DNA, or degradation of the viral RNA upon infection, or RNA-RNA duplex induced degradation of target RNA? So you can go through this. So this is what we are talking about. If you look at the illustration very with a lot of attention, very attentively, then maybe you will be able to do it very normally. Right? It is not that difficult, but this particular illustration is something that is a little confusing. Okay, so we'll come to the answer. Okay, my slide is frozen. Yeah. So the answer for this particular question is that this particular highlighted red triangle, what it represents is RNA duplex induced degradation of the targeted RNA. Now, at some point, it might look like we have already reached amino acids over here, right? It is written amino acids and certain amino acid codes have been given. But if you actually look at what is given is, below those amino acids is a sequence that is given. And this is a sequence that is basically an mRNA sequence, right? Or an RNA sequence. And what is being introduced into this machinery is an shRNA. So what is a shRNA? It is a short hairpin RNA. What does a short hairpin RNA do? It basically is responsible for RNA interference, right? So it is a synthetically made compound which will basically go and interfere with the RNA. So if there is introduction of this shRNA itself, what will it bind to? It will bind to the RNA. So over here, the RNA sequence that was depicted earlier, it has shown to be degraded over here, right? How? Because this particular red colored sequence has come and bound with this. So now the protein that it was supposed to make, it will not be able to make that. All right. So moving ahead. Okay, so I have also kept this illustration. The reason for that was basically telling you that mRNA, which comes out of the nucleus, can be targeted by any such RNA. shRNA was one that we talked about, which was given in this illustration. The other two naturally occurring double-stranded RNAs that are present in the natural environment themselves and sometimes in our own cells are siRNA, which is small interfering RNA, and miRNA or microRNA. So these are also already present in the system, and sometimes this degradation could occur due to these endogenously expressed RNA interfering molecules instead of the exogenously expressed shRNA, right, that we talk about. So coming to our fourth question, which is, which of the following statements correctly identifies the difference between global knockout and conditional knockout? So this, I think, is something that might have been very clear to you by now. Sir basically talked a lot about it. For people who did attend my last session, I also extensively talked about the difference, particularly between global and conditional knockout. So let us go through the options. A global knockout mouse can be used to study all types of genetic disorders, whereas a conditional knockout mouse can be used to study only metabolic disorders. Or a global knockout mouse has a gene deletion in a tissue-specific manner, whereas in a conditional knockout mouse, one can delete a specific gene at a specific time point. Or a global knockout mouse has a gene deletion in all the tissues, whereas a conditional knockout mouse has a gene deletion in a tissue-specific manner. Or the final one, which says global knockout mouse can be used to create a deletion of a gene, while conditional knockout mouse can be used to create a knock-in mouse model only. So I do option what?
C.
Yeah, okay. So that is actually correct. This is basically the definition of these particular mouse models. This is how they have been named, right? Global means that all tissues, in all the tissues, gene deletion would occur, right? While conditional means that we can condition it, or we can basically put a condition that, okay, I just want astrocyte specific gene knockout, or I just want kidney specific gene knockout. So this is where a tissue specific gene deletion can happen, right? Again, I have brought in the same illustration from last time, which basically has these two represented. So a global knockout is also known as a constitutive knockout model. So sometimes you might hear this term instead of global knockout. So that is what it means. It basically means a global knockout mouse, right? So in a constitutive or a global knockout mouse, what will happen? There will be a tissue-wide, like all over the organism-wide deletion of the gene of interest, right? Whereas a tissue-specific knockout or a conditional knockout, what will happen is that, for example, over here, in this particular hypothetical experiment, people were targeting tendons. So only from the tendons, the gene of interest will be lost, because the promoter that will be applied before this particular construct will contain a tendon specific promoter, right? But in the rest of the body, since that tendon specific proteins or regulator elements will not be there, it will not get knocked out, right? Okay. Yeah. All right. So moving ahead, number 5 is the genetic editing tool CRISPR-Cas9 uses a guide RNA, right? The guide RNA is a RNA sequence that helps in identifying the target DNA locus where it makes a double-stranded break, or a RNA protein complex that anchors the endonucleases to the target DNA, or a RNA protein dimer that helps in formation of the Cas9 complex, or an RNA protein complex that guides the restriction endonuclease to identify the target DNA sequence. So which one do you think is the function of the guide RNA? Which one?
I think D.
You think B? Okay, because our voice is overlapping. That is why I'm having to ask you again every time. So sorry for that. So you think that it will be RNA protein complex that anchors endonucleases to the target DNA. So that unfortunately is the incorrect answer. The correct one is the first one. So what a guide RNA will do is it will be a sequence that will be, sorry for that. It will be a sequence that will be a part of the CRISPR gene itself. And what this sequence will do is that it will basically be homologous, or not homologous, it will be complementary to whatever sequence we actually want to target. So what it will do is that it will identify the target DNA locus, because it will be the complementary sequence to that, right? So that is how it guides, because it will take the entire complex and it will guide it to the DNA locus, right? So I think I might have put an illustration for this. Yeah. So this is how or what a CRISPR basically sequence looks like. So what this can be, I'm so sorry. So what this sequence can be divided is into two parts. The first is target specific cRNA sequence, and the second is the tracer RNA sequence, right? So what this first sequence will do is, which this is also known as the guide sequence, it will go and it will bind to the DNA. What this tracer sequence will do is that it will later recruit the Cas9 complex to come and bind over here, right? So if you look at this particular illustration, you can see that this gray represents the DNA. This part of CRISPR, which is the guide RNA or the target specific cRNA sequence, will go and bind to some part of the DNA from which it is complementary. And what this tracer RNA will do is that it will recruit this Cas9 and it will help in basically creating the nicks in the strand, right? So I hope that concept is clear.
Madam, what is the difference between A and D was only RNA sequence and RNA protein, I think. Otherwise, both options are same.
Yeah, so but it does not have any protein component in it. Right? When you are talking, for example, if I talk about a RNA DNA duplex versus an RNA complex or a DNA complex, it will be two different things. It will be very different because molecularly they will be treated by the cell in a very different fashion, right? So over here, this is purely an RNA sequence. It does not have any protein complex in it, right? Later, it will
RNA
go
is
and
not
recruit
protein. Okay.
some part of the protein. Yeah. Also, you said that A and B only had this difference, or A and D only had this difference. That is what you said. Yeah, A and D are more similar, but it does make a difference when you are talking about RNAs, DNAs, and proteins. You have to keep in mind what particular biomolecules are actually involved, right? So in the sixth question, we ask, which one of the following options is a true statement for homologous recombination process? So the first option is it a type of genetic recombination in which nucleotide sequences are exchanged between two similar or identical molecules of DNA? Or it occurs during the formation of egg and sperm cells? Or it mostly occurs between homologous chromosomes bearing distinct markers surrounding the exchange region? Or there is another option, which is all of these. So homologous recombination is something that we have been familiar with almost since class 12th, right? So there we were first introduced with this concept of heterologous and homologous genes or chromosomes, sorry, not genes, homologous chromosomes, right? So we are kind of familiar with this concept since after class 12th. It's
A.
what? It's option A. Are you sure? Okay. So we will go through this.
Either A or all of these.
Yeah, so it is all of these actually, right? So why is it all of these? When we talk about homologous chromosomes, when do we have homologous chromosomes? When there is this process of meiosis that is going on, right? So when does that process of meiosis occur? When we have to basically make the gametes. The gametes in humans are the eggs in case of females and the sperm cells in case of the males, right? So, firstly, okay, let us look at the homologous chromosomes first. So, this particular set is a homologous chromosome, right? So, they are similar and they are two copies of the same chromosome, kind of. And if we look at this chromosome, can we point out what are sister chromatids and what are non-sister chromatids? Can anybody do that for me? Just asking. I will still give the answer. Okay. So over here, if we look at this particular illustration, the one in green, both these processes in green are sister chromatids among themselves, and these processes in purple are another set of sister chromatids. But if we talk about one arm of this green and one arm of this purple, then they together cannot be called sister chromatids. They are non-sister chromatids in a homologous chromosome. So this is one concept that I think it hadn't been very clearly explained to us when we were younger. So I have across my graduation, post-graduation also had a lot of classmates who have had a lot of problem with what this particular homologous chromosomes are. So I will just take one moment to explain that also. So if we talk about, oh, it is unresponsive. Let me just move this. Please forgive the drawing because I'm working with a mouse over here, not mice. So, for example, I drew one chromosome over here, and with another color, I will draw another chromosome over here, right? So this is another chromosome. Now, please assume that both of them are of the same size. Only then can we call it homologous chromosome. So since I was doing a freehand drawing, I could not make it the same size. But that is what we will assume, right? That they are exactly similar in size, right? So what will happen is that this particular chromosome has two chromatids, right? One chromatid and the second chromatid. Similarly, this one also has two chromatids, first chromatid and second chromatid. Between these two, they'll be called sister chromatids, right? They will be known as sister chromatids. And the red ones among themselves will also be known as sister chromatids. But if we talk about, let me draw the red ones also outside. So this is one sister chromatid and this is another sister chromatid of this red chromosome, right? If we take one of this red and one of this orange, these together cannot be called sister chromatids. They are known as non-sister chromatids. Although these chromosomes together will be known as homologous chromosomes, right? So if there is any problem with this concept, please interrupt and ask. I am completely okay with that. But I hope that I have been able to put it across properly. So if we talk about homologous recombination process, so when does this homologous recombination process take place? It takes place usually during meiosis. When does meiosis occur? During the formation of eggs and sperms, right? And usually this
Madam, it is
is
egg or ovum?
yeah.
It is egg
Huh?
or ovum? It is egg
Yeah,
or
so you
ovum.
can call it either. You can call it either. So that is just a terminology. In humans itself, we call it as eggs also, right?
Because eggs will
So
be mostly called it is after fertilization. That's why I was confused. Okay.
okay, yeah. So again, that is very technical, but it is true actually. So there are various stages of the ovum also, right? So that is why over here, I think it has been listed as egg in this particular context because ovum could either be mature or premature. And there are some stages in the ovum wherein meiosis has not yet occurred, right? So maybe to like do off with that other perception that people might have, like there are some phases in oocytes themselves wherein meiosis has not yet occurred, right? So maybe to not leave ambiguity in that phase, egg has been mentioned, but yes, ovum could be an option. If you are again not confused with the concept of it is a process after meiosis. That is what we are trying to say over here. So egg, usually in eggs, in case of humans, meiosis is actually completed after fertilization. I think you might be aware of this concept. Maybe to remove that ambiguity, egg has been mentioned over here. But if I start explaining that, this will become a physiology class. So maybe first you will go and check it. And if there is any doubt, I will be happy to explain it next day. So the first option is obviously correct because that was another guess that we were getting, right? That A. It is a type of genetic recombination wherein nucleotide sequences are exchanged between two similar or identical molecules of DNA, right? So that is why I was asking you to basically assume that these are two similar sized DNA chromosomes, right? So they are similar and there is some genetic material that is getting exchanged between them. It is occurring in eggs and sperm cells also. Also, this third one could be something new that you learned today, that it mostly occurs between homologous chromosomes bearing distinct markers surrounding the exchange region. So you might have studied it earlier sometime that there are some regions in the chromosome that are more vulnerable to crossing over or recombination than other regions, right? This is what this point is trying to say basically. So that is it. The answer for this particular question was all of these. And if I may ask, just to for you guys to basically dig back into your brains, when does recombination occur in meiosis? Which meiosis and then which phase in that? Could anybody recall that? Just for information's sake. Okay, so it occurs during meiosis 1. So there are two phases of meiosis 1 and meiosis 2. So it occurs during meiosis 1. In that, during prophase 1, and in prophase particularly, this happens during Pachyt, right? So this we study when we study cell division, right? So over here, we were just talking about it for a brief knowledge upon the subject, but we can move on to the next question, which is, which one of the following most appropriately describes the classical central dogma of biology? So this one I think is the easiest question of all. The first option is protein to RNA to DNA. The second is RNA to protein to DNA. The third is DNA to RNA to protein. And the fourth is DNA to protein to mRNA. Yeah.
C is correct, but instead of RNA, they should have written mRNA.
mRNA, right? But RNA is also one of the intermediate phases. Don't you agree? mRNA is a successor of RNA itself. You are correct. Even if they would have written mRNA, it would have been fine. But even if it is RNA written over here, it's still fine because RNA precedes mRNA, right? DNA is directly transcribed into RNA. And that after post-transcriptional modifications becomes mRNA. So this is also an intermediary process. And mRNA is also an intermediary process. So either way, it would have been correct. So the central dogma of molecular biology is basically a theory that states that genetic information flows only in one direction. That is from DNA to RNA to protein or RNA directly into the protein. This happens in case of viruses, right? Okay. So just for the sake of representation, if this was a DNA, it would have been transcribed into an RNA or an mRNA, which would have been a mature form of that particular RNA, which would later be translated into a protein. So this was the central dogma of biology. So can somebody tell me who gave the central dogma? If not, I have a slide on that during my concept in my other presentation. So we'll discuss that. So I'll leave that question right here. So the eighth one is, which one of the following approaches cannot be used to study the transcriptome? RNA-seq, microarray, RT-PCR, or pedigree analysis. Which one of these cannot be used to study transcriptome? So I think three of these you have come across during this week's lecture. One is very obviously what you have not studied during your transcriptomic conversation, which is pedigree analysis, right? When did you last study pedigree analysis? I think in week one's lecture, when we were talking about positional cloning, right? So that pedigree analysis basically precedes positional cloning. Okay.
Madam, central dogma was given by, I think, Francis Crick or Watson.
Yeah. So it was given by Francis Crick.
In
so
his
both of
lecture,
them together had Yeah.
In his biology lecture, he has given the first time he has introduced that central dogma in the biology
Yeah.
lecture in his class.
Oh, that
He
I'm
was a,
unaware of.
he was a teacher, and first time he has
Yeah,
introduced
yeah.
that it in his own class.
Oh, that is actually a new information for me as well. So, yeah, it was Crick who did introduce central dogma, and later both of them together they discovered the structure of the right? Now, question. Um, just for a moment.
Voice problem, madam.
Yeah. Actually, somebody's mic was left on. I have muted the respective person. So I will share my slide again. Yeah. So we are at the penultimate question for the previous year question part, which is which one of the following option correctly identifies the stepwise process in positional cloning approach? So this is something that was dealt in the last week's lecture. So it will just test your recollection capabilities. So is it genetic mapping, transcript mapping to gene sequencing to physical mapping, or genetic mapping to physical mapping to transcript mapping to gene sequencing, or physical mapping to transcript mapping to gene sequencing to genetic mapping, or the final one, which is genetic mapping to transcript mapping to physical mapping to gene sequencing? Which one among these do you think or do you recall is the correct stepwise process in positional cloning? So in this, I
B.
would like to wait for an answer. Yeah, so B is actually the correct answer. During the positional cloning, we saw that pedigree analysis is what basically helps in genetic mapping, right? It helps to point out a particular chromosome or a particular locus in a chromosome, right? So this is that part, the genetic mapping. Later, it helps identify the minimal disease region, which is basically this region, which is further mapped physically to identify the gene, right? So further physical mapping is done to identify a particular gene. Later, this gene is looked for expression analysis using transcript mapping. Later, transcript mapping is done to look for expression of this gene, and it is further sequenced to identify what particular mutation was actually causing this defect, right? For example, over here, this G has been highlighted, so it would have been a single nucleotide, maybe. Oh, I have.
Cystic fibrosis.
Yeah, so this was an example of cystic fibrosis. I am unable to recall what a this particular side-directed mutagenesis is called, like the type I was unable to recall. So, okay, if that comes to me, I'll come back to this slide. Yeah, so it was an example of cystic fibrosis actually. The final question is, genetic manipulation in mouse models can help us to understand redundant gene functions, gene mutations that are embryonically lethal, disease pathology, or all of these. So genetic manipulation, in what approach did we actually do genetic manipulation? Was it forward genetics or reverse genetics?
D. Not D, madam. D.
The
This is
answer is
pathological
D
all of
here. All of these is the correct answer. Now I have another question regarding this. In which one of the approaches, forward or reverse genetics, did we actually perform gene manipulation?
This
Was
is
it
the
forward?
example of reverse genetic, madam.
Yeah, so it was
And
reverse
the
genetics.
positional cloning was an example of forward genetics.
Yeah, yes. So, yes. So if we are manipulating any genome, or we are actually like manifesting a mutation in a particular genome, what will happen is that it will help us study the redundant gene functions, right? Why? Because in case of forward genetics, maybe because this the function of this gene is taken up by another gene. So we are unable to actually see what kind of a function it performs, right? Or when it is not there, what will happen? We will never be able to identify this gene altogether. In case of embryonic lethal mutations, that particular mutation will not live ever in an organism so that we can identify it, and it will also help us study disease pathology, right? So that is the end of the first part. So we'll move on to the second presentation. I will just plug in my laptop. Please give me a moment. Yeah, so please give me a moment. I'll just plug in the laptop. I would like to take a moment. My laptop has heated up terribly. I just want to bring in a stand. Please excuse me for 1 minute. Okay. So I hope I'm still visible. Yeah. So I'll share my screen for the next presentation. Uh, yeah. So people who did join late, how I had planned to go about this particular lecture was first by discussing the previous year's question for the particular week that you are in and the particular assignment that is due for you, so that those concepts could help you maybe solve this year's assignment as well. And after that, the point that we have presently come to, which is basically to discuss some extra questions and some extra concepts. And that is what we are going to start right now. So for this part of the presentation, let us begin with what functional genomics is. So yes, last time, since the lectures hadn't really come like they hadn't full-fledgedly talked about what functional genomics is, I had not yet included the actual definition. We had just talked about what is genes, what are genetics, what is the difference between genetics and genomics, right? So this lecture, I would like to begin with basically discussing what is actually functional genomics. So functional genomics is basically a study that attempts to determine the function of all gene products encoded by the genome of an organism. Also, it involves the study of how genes are and intergenic regions of the genome contribute to different biological processes, right? Okay, from this, I remembered an instance that I was going through the discussion forum, and there I really do not recall who, but somebody had basically asked as a query for the previous year, for the previous week's assignment that you guys had submitted, uh, uh, that genome, whether it should include the microbiome also, right? So, and in that, there was actually a line that I read that we had discussed it with the TA, and this is what they had basically taught us. So, since there are two TAs in the course, I do not know which one the person was talking about, but from my side, I would like to clarify that genome is basically the genetic material of a particular organism, right? When we discuss about metagenome, which is also what I talked about in my previous lecture, metagenome basically includes the genome of the organism and its environment. So there, uh, that particular concept would also include the microbiome that is present inside the organism. But just genome will not include that particular aspect, right? So that was just one clarification I wanted to make. So I hope this is a very simple definition. I hope it got across to you. Let us just look at how basically this concept could be performed or it could be studied, right? So what is the main aim of functional genomics? If we talk about the main aim,
Excuse me, madam. Hello.
Yeah.
It
Yeah.
is me, Balaji, who
Just a
put
moment,
that
let me-
question on the
Oh,
program.
okay.
Because
Yeah.
when we discussed, you mentioned, yes, it will also include the microbe. Because the question, particular question they said is
Yeah.
genome means only chromosome. Genome
Yeah.
does not include the what is that, mitochondrial genome. And then genome includes all the chromosome and then any other microbes associated with that. That was the three options we had. So
Okay.
we cannot say genome is only chromosome because it also should include the mitochondrial genome, but it did not have that option. That's why I was confused. And then I had that question asked to you, and then you mentioned not metagenomics came later, but you said that yes, we should include the microbes or whatever is available also for that total organism. So that's how I lost 1 mark. Does not matter, but
Yeah.
just wanted to make a point to you. Thank you again. Clarification is there now. I'm not worried about the marks, but definitely we got some clarification. But that question formation was wrong in the sense it did not include the uh, mitochondrial, DNA, which they should have included. Only they said the chromosome only is genome. That is wrong. That question is not formulated properly. Thank you, madam.
Actually, I found that problem with a lot of previous year questions also. So that we will try to communicate to these people at NPTEL. Yes, Ajay.
Probably in chromosome sequencing, we don't, in genome sequencing, we don't sequence mitochondrial DNA because
No. We
mitochondrial
just
DNA is not
go for
no,
Yeah.
it is not our own DNA. It is some parasites, which is means symbiotic parasites, which is helping to make energy more. That's why in genome sequencing, we are not including mitochondrial DNA. That's why the correct answer will be without mitochondrial DNA. Means genome sequencing is only for chromosome. So genome includes only chromosome. When you say whole chromosome, then metachromosome, that's what at that time we should include chromosome and all the chromosomal microbiomes that is included, including mitochondrial DNA. For pedigree analysis, to trace out our generation back, who is our mother or who is our father, that's very easy to trace from mitochondrial DNA because it only comes from the mother, and Y chromosome only comes from the father. So for tracing purpose, mitochondrial DNA is used, but for genome sequencing, we don't use mitochondrial DNA because we think it is some parasite which is engulfed in the human chromosome. So if we think parasites, so all other parasites also there.
Right? First of all, it is not a parasite. It is a symbiont theory. And it is a theory. So we can't really say that what we have is a symbiont, right? What the theory is that long time ago, when the eukaryotes were basically being developed, at that time, some kind of a bacterial symbiont. It came inside and it continued to reside within the eukaryotic cell because it helped in energy formation and supply to this particular eukaryotic cell. And this particular eukaryotic cell was a good host for it. It provided a good environment for this particular symbiont to live in. It was a safe environment, right? So it is a symbiont theory, but it was how basically mitochondrial DNA evolved, or how mitochondria evolved, right? We cannot generalize that statement to us right now, right? We cannot say that it is a symbiont that is living within our cells. For us, it is a cellular organelle. Okay.
So
But
when
the
I,
rest of it was
yeah.
true. Yeah.
When I
Yeah.
spoke to Professor Ganesh
Yeah.
on this topic, he said that mitochondrial DNA is also part of the genome of the organism. That's what he
It
said.
is that.
So
That is
therefore,
actually my understanding also.
this
But
question is
yeah.
a little bit needs clarity, and see what can be done on that question, that particular
Yes,
question.
yes.
Thank you.
Yeah. Oh. So I would have to check if my screen is still visible to you guys, or should I share it again?
The screen is not visible, madam.
Okay, so I'll share it again in that case, right?
Now it not it was visible. It
Just
was
a moment.
visible close.
Yeah. So I hope we have all come back to the presentation, right? So in this presentation, we were basically saying what functional genomics is. So the major goal, if we want to say it in one line, what is the goal of functional genomics? It basically means to assign a phenotype to a genotype, right? So we have a gene. We want to see what it does. So to assign a phenotype to a genotype is what is the aim of functional genomics, or what is the ultimate goal of functional genomics, right? So this phenotype has to be found out. So for this, there are multiple approaches that have been developed. Earlier, when genomics came in, we just used to sequence the DNA, right? So when we just used to sequence the DNA, we used to call it genomics, right? Later on, multiple techniques basically developed to further the study of functional genomics. And these include epigenomics, which is to study the epigenetic molecules that are present inside our genome. Transcriptomics, which is the study of transcripts in our basically in our body or in our cell, right? So what are these transcripts? They are basically mature RNAs or mRNAs. Also, there evolved other fields such as proteomics, which is the study of proteins, the entire repertoire of proteins that are present within us, and the study of metabolomics, which basically means that the study of the entire repertoire of metabolites that are present within our body, right? Now these fields have diverged even further. There are multiple more kind of omic techniques, like lipidomics is one such technique that is coming up, that lipids have to be studied, the entire lipid biomolecules that are present in our body, they have to be studied, and how each of those lipids might affect the phenotype. So these kinds of studies are ongoing, and that is why sometimes we refer to this particular decade as the omics era, right? So omics is on the rise, or at least in the past decade, it has been on the rise. So all these high throughput omics techniques have been going on majorly, right, in the past decade. So that was the introduction of functional genomics. Now coming up to one of the questions that was asked in the previous lecture. I believe Balaji was one of the people who asked the questions. Is there somebody trying to join again? Yeah. So I will share my screen again. Yeah. So there were two people actually in the previous class. I couldn't recall the other person's name. Maybe she's in the class. No, she isn't. So if I saw the name, I would have remembered. But since I am not seeing the name, it is hard for me to recall. So there were these two active people in the previous class, and what they wanted to ask was that during splicing, we discussed alternative splicing last week, and during splicing, is it possible? We talk about alternative splicing. Like what is alternative splicing? Is that for example, it is not important that if 5 exons are present, then all 5 of them will be there in one mRNA, right? From one sequence of DNA, there could be, or from one gene, which has 5 exons, there could be multiple. Okay. Please excuse me, guys, because I'm getting this notification, which also I need to cater. Okay. Sorry. So it is basically a mechanism wherein our machinery is very smart because it can use one gene to make up several proteins, right? How can it do that is, for example, there is one gene. It has 5 exons, right? 5 coding regions. So it does not really need all 5. It might need all 5 for one kind of protein to be formed. It might need 3 out of those 5 for another kind of protein to be formed, and so on. So there are multiple permutations that could be, and combinations that could be applied to these 5 exons, right? And our cell very well utilizes that, so that we have to use less number of genes for multiple protein production, right? So that is what alternative splicing is. For example, it could happen that exon 1, exon 3 and exon 4 will be present in one particular mRNA, and that will form one particular protein, say insulin, right? For example, if exon 1, 3, 4, and 5 are present, say that it will form galactase instead of insulin. So multiple different kinds of proteins can be formed, which is one of the advantages of splicing or alternative splicing for that matter. But the question that we landed on last time was whether the order of these exons can change. So we say that this way the order can change. Instead of 1, 2, 3, 4, 5, we can have 1, 2, 4, 5. We can have 1, 2, 3, 5. But we might be able to have 2, 3, 4, right? So that way the order can change. But the question was, can it happen, or is it possible that exon 4 comes before exon 3, or exon 2 comes after exon 5? Is that possible at all? So this is one question that I was also very confused about. So I went back, I checked. So up to my knowledge, this is something that is not possible. But for this, I basically came up with why logically it wouldn't be possible. So for this, I would like to introduce one more concept over here, right? So, okay. Yeah, sorry. So for this, to completely solve this problem, yeah, Ajay.
Madam, in alternative splicing,
Hmm.
exon A, exon B, exon C, exon D, whatever you had said, exon 1,
Yeah,
exon 2, exon 3, exon 4, the part
yeah,
between exon 1 and exon 2, can it be a
yeah.
part of mRNA in alternative splicing?
No, introns cannot be a part of mRNAs at all. But this is again one interesting concept that I dealt with last time. But it is not always okay. I will not confuse you right now. Maybe I will show that slide at the end of this particular discussion. But for now, what we will say is that introns cannot be a part of mRNAs at all. Okay.
Just
So,
madam,
for
just
example,
just if we change the definition of exon and intron, whatever
yeah,
remains inside the nucleus after splicing is intron. Whatever goes outside the nucleus in the cytoplasm, that is exon. So it will be the part between exon 1 and exon 2 written can be part of alternative splicing. That's what I got from the alternative splicing lecture from Professor Ganesh. It can be a part
Yeah.
because
So
introns is a part
that's,
which is not present in mRNA.
yeah.
Intron, we decide intron after mRNA is formed, not before mRNA is formed.
Before
Intron
mRNA,
exons.
it is a part of the RNA chain that is present. It is just not the part of the matured form of that mRNA that is going to go and be involved in translation. I am unable to get your question. Can you please frame it again?
Madam, mature RNA form in the nucleus or outside the nucleus?
It is
Alternative
formed
splicing,
inside the
splicing is
nucleus.
taking inside the nucleus or outside the nucleus?
Inside.
So then my concept is clear. Whatever the part written in mature mRNA, that is exon. Whatever the part left out of mature mRNA is intron. So the part between this, whatever we cannot pinpoint,
That
this is
is
the
not
exon, this is the
actually how
intron.
we define introns and exons, because it is not important that the part that is spliced out, it will remain inside the nucleus, right? First of all, let us first correct our definition of how we will define an intron and how we will define an exon. So what an exon is, a protein coding region, okay? An intron is a non-protein coding region. It might have other function, right? But it is a non-protein coding region. So since it is not coding for a protein, it will not be a part of mRNA. But it is not important that it will stay in the nucleus, right? What happens to it is actually one of the questions that we have not been able to solve universally, right? So does that clear some part of your doubt?
Translated UTR region is also part of RNA. They are
Exon.
not coding.
UTR is a part of the exon. Okay, it is also an untranslated region, but it is a part of exon. Why is it a part of the exon, or why is it a part of the mRNA? Is because this UTR region is what will basically be the basis for where the 5' end and the poly A tail will come and attach, right? And they are important for the mRNA to basically go out of the nucleus and into the cytosol, and in the cytosol itself also it prevents degradation of this mRNA, right? These ends, the 5' cap and the poly A tail. So they help in prevention of degradation of the mRNA. So since the UTR is basically helping in that, we still consider them a part of the mRNA. But introns, they are not a part of the mRNA at all.
Whatever we make mRNA, it is with the exon only. That's what my thinking is. Maybe I wrote.
Yeah.
And whatever is left out, that is intron. So
That is introns.
so before splicing, we cannot decide in pre-mRNA this is intron or this is exon.
No. Again, it is not what is left out is introns. There are regions that are either coding for proteins or not coding for proteins. As in, for example, if we talk about alternative splicing also, over here, for example, if you see in this particular middle section, right? Over here, we see that exon 1, 2, 4, and 5 are mentioned. Exon 3 is not mentioned, right? So in this particular mRNA, exon 3 is not present. So it has also been spliced out. But it is not an exon just because it has been spliced out, right? Because in other mRNAs, it might be present, right? And it is basically responsible for forming a protein. So we cannot say that whatever is spliced out is introns. But like, for example, we cannot, there are some things that, let me just think of a very simplistic example to give. Okay, for example, if we say that Earth is our home, it is something that is acceptable. But if we say that we are Earth's home, that is not acceptable. Similarly, if we say that introns are spliced out, that is acceptable. But if we say that whatever is spliced out are introns, that is not true. Can you relate to that? There are a lot of such examples actually. Yeah. Okay, just sit on it for a while. If it still persists, maybe I can spend 5-10 more minutes after the class so that for everybody it is not hampered, right? So we'll discuss it after the end of this lecture, maybe. Okay.
Okay,
I will try to convince you further. Okay. So yeah, we were talking about why basically the order of exons cannot be changed, right? For this, I was going to introduce a new concept, which is how basically introns are spliced out. So introns are spliced out using machineries which are known as spliceosomes, right? So they are basically multiple molecules that come together. They form a complex which is known as spliceosome. And what the spliceosome does is that it basically cuts in between and takes the intron out or excises the introns, and the exons come together, right? So how do spliceosomes recognize introns? That was my first question. So if you look at this particular sequence, can you see anything that is common in that? So for example, consider this as exon 1, this as exon 2, this as exon 3, and this as exon 4. So sequentially, we have 4 exons, right? Is there something very unique that you see over here?
Mostly AG or GT sequencing. It is most of the time
Yeah,
G is common. G
yeah.
nucleotide.
That is exactly what I wanted to show over here, that these sequences at the end of each exon are similar, and at the beginning of each exon are also similar, right? So this is GT generally at the end of every exon, and AG generally at the beginning of every other exon, right? So actually the spliceosome, which consists of many molecules, which we are not going to go in detail of, will come and recognize these sequences, which are GT. In case of RNA, it will be known as GU instead of GT, right? Because uracil is what will be present in RNAs, and thymine is what will be present in case of DNA, right? So over here, GU and AG are the sequences that these spliceosomes come and they recognize. And then what do? They basically make a cut, right? They make a cut. So what happens is, for example, these are 2 exons, and in between we have, I hope I'm visible to everybody, right?
Ma'am,
Okay,
can you
just,
present the screen again?
yeah. I thought I might not be visible. Which, okay, I hope everybody could follow me till here. Okay, so what we said was that GT, AG are sequences that are common at the end of and beginning of respective exons, right? So what the spliceosome does is that the spliceosome comes and it recognizes these particular sequences, GU and AG. And after recognizing these particular sequences, what it does is that it makes a cut over there, right? When it will make a cut, what will happen is that first we had 2 exons, right? And this part wherein nothing is present, that is basically an intron. When it will recognize these 2 basically borders of the exon, so what it will do is that it will basically try to bring these 2 sequences together, right? So when it brings these 2 sequences together, this particular area will form a loop, right? For example, if I already have a torn page, I will try to explain. Yeah, I do. So for example, if these 2 sequences were to come together, the middle sequence, which is the intron, will be formed as a loop, right? After that, this loop will be excised and the 2 introns will come together, right? Because this loop is getting formed and it is being excised. It is not like there are cuts happening over here and here. This will be cut and this will be cut and this will be cut, and then they will be stitched together. This is not how it happens. It sequentially forms a loop, and the two exons that are lying adjacent are brought together, right? So this is the GUAG rule, that the first two and last two dinucleotides are basically recognized, right? And the region from the middle is removed. So since this is how splicing occurs, it is not possible that, going back to the previous slide, that exon 1 can come after exon 4, right? Because after exon 4, exon 5 will only be present, right? If the fifth one is not getting spliced into an mRNA, and if it is being spliced out, then maybe the sixth one can come and join, but it will not get jumbled, right? It will shuffle, but it will not get jumbled. I hope that this concept is clear, because it was one of the questions that we had left since the previous class. Okay, so we'll move on to the next slide, which is basically what is the current use of genetics at the clinical level? Again, this was one of the things that we talked about very briefly in the previous session. And I have tried to put an overview of how basically genes are getting recognized and diseases, genetic diseases are getting recognized these days at clinical levels. But obviously there are a lot of technical things that I have avoided to keep over here, because then there would be 10 more terms that you will have to understand, right? But I have kept at least, I think, 6-7 slides for this particular topic. So this is what I'll be talking about. Please excuse me while I have a water break. Okay. So what is the current use of genetics at the level of clinics? So that is that actually in hospitals, in clinical settings, how is genetics being used? So first of all, it is being used to understand diseases, right? So to understand diseases, what does that mean? For example, if we have a patient, right, and we want to see that suppose we have a patient with symptoms that are not really matching any particular disease. We have done pathological studies also, and they are also very ambiguous. They might be resembling some kind of a disease, but maybe they are not fulfilling all the criteria for those diseases as well, right? So we could think about two possibilities. For example, maybe that person has a lot of symptoms that are matching maybe a classic Alzheimer's disease patient, but a few major criteria are not being fulfilled, right? So either we can think of it as being a subtype of that particular disease, maybe just some kind of a subtype of Alzheimer's disease that we have not yet discovered, right? Because it is always possible to have rare occurrences in these cases, right? Because there are a lot of genes that could go haywire. And the second possibility could be that it is a novel disease altogether. And for that, we need a lot of, for both the notions, we actually need a lot of work or research to actually understand what is happening over here, right? So what we will do is that probably at that time, if the symptoms of that particular person are not matching the symptoms that are already understood by us, or some gene defects that are already basically verified by researchers and doctors, right? So we might not be able to immediately help that person. But in the long term, what our goal is to basically collect such data to make it publicly available, so that if at all a similar symptoms occur in a patient in, say, Israel or in, say, US, right? So what will happen is that we will be able to basically get their files. We will be able to get in touch with the physicians or clinicians that are treating them, and we might be able to discuss that, okay, this might be what possibly is causing this particular disorder. Is it something new that we are seeing, or is it just a modification of some other previously understood disorder that we already know about, right? So that is one thing, that disease understanding is very important, and it is currently going on both at research level as well as the clinical level. Now, another interesting thing that genetics is used for is disease diagnosis. So, how disease can be diagnosed based on genetic screening is basically, so these are two very interlinked terms, genetic screening and disease diagnosis. So, for diseases to be diagnosed, maybe a patient comes in, and what we say, ask that patient to do is to provide us with a sample that we will send for sequencing. Right. Once that sequence comes back, we can basically check what kind of genes are mutated in that particular individual, and if that mutation is already known to occur in a particular kind of disease. Right. So, for example, if that mutation was expansion of the CAG repeats, right? There are a lot of CAG repeats that are present in that particular patient. So we can basically say that, okay, so we think, and we have actually evidence through that sequencing, that it is Huntington's disease that you are looking at. And at that stage, it might not have been that identifiable in that person. But if the genetic sequencing is done immediately, and if at an earlier age actually we are able to understand what that particular disease is, maybe even if we can't stop that disease altogether, maybe we might be able to delay its progression, or we might be able to like increase the life expectancy or the quality of life of that person, right? Then comes genetic screening. So what genetic screening basically means, it is very interlinked with diagnosis. Over here, what we say is that the family history of a person, because if we have a family history of a person or a pedigree that we can make, we can basically see that whether a trait or a symptom or a particular disease or disorder, whether it has occurred in other individuals. Probably if it has occurred in other individuals, it is genetic in origin, right? And then maybe we can be able to identify locus and do that candidate gene selection through traditional cloning. All these things might be possible, right? The final and broader aspect that we are looking at now is gene therapy, right? So if we understand the disease genes, and if we understand what they do, right, at all levels, we have this understanding very well established that this is a disease, it is caused by this gene. This gene originally, physiologically, when it is normal, this is what it does. But once it becomes abnormal, it is causing this particular defect, right? So once we have this entire understanding, we can say that, okay, I think that if I am able to somehow induce the function of this gene within this said individual who is lacking this gene, I might be able to make him better or make her better. Like, for example, if a person has some uh, enzyme deficiency, right? And that enzyme deficiency is stemming from a particular gene, right? It is not a polygenic disorder, it is a monogenic disorder. There is one sole gene that is responsible for the deficiency of an enzyme. And if say I was able to basically put a promoter site adjacent to that gene so that it is transcriptionally active, I would be able to rescue this deficiency in that person. That kind of these studies and research is what is gene therapy. And now there are clinical trials also in case of some diseases that is going on for gene therapy itself, right? So these are what currently genetics is being used for at clinical level. Now, if we see how basically we can diagnose a genetic disease. So this was one of the questions that was asked, right? So for this, first of all, I would like to recommend a book that I found at NCBI's National Library of Medicine. This book is basically named Understanding Genetics, and the entire name is a District of Columbia Guide for Patients and Health Professionals, right? It is a freely available book, and maybe if you type these references, you'll be able to get a link to it. And over here, they have actually, there's an organization called as Genetic Alliance, if I'm not wrong. They have basically contributed in formulating this book. And there are multiple chapters of how we can understand genetics, what do genetics lead to, what kinds of diseases they cause, how we can diagnose them, what are, like, it is a very detailed version of what I'm going to explain right now, right? But for people who are interested, you must go back and look into this also. So, if from this, from chapter 2, that is another reference I'm giving to you, I have picked up diagnosis of a genetic disease. So over here, they have basically told us that there are some diagnosis methods that are involved in diagnosing a genetic disease at a clinical level. What are these methods? They are physical examination of the patient, detailed family history of the patient, which is actually very nice if you can obtain it in case of a genetic disease particularly, because it gives you a lot of hints and clues, right? Or, and clinical and laboratory tests, right? So from this, we can basically ask a question about when do we suspect that diseases have a genetic cause? So when at all, there are so many diseases that exist. Why should we think that a particular disease is genetic in nature or not genetic in nature? So there are certain things that we can look out for. So say you were a clinician and you had a patient. How would you determine whether you should delve into this genetic side or whether you should look at the pathology or the physiology that is that person is getting affected by? Which one should you do first, right? So, for example, if your family members that were listed in the family history have severe disease, similar disease conditions, so that would be a very good indicator of it is a hereditary disease and that has genetically been transmitted, right? Something like that. It might be possible. Also, if the family history of common adult conditions such as heart disease, cancer, dementia occur in 2 or more relatives that are relatively younger in age, it might also suggest a genetic predisposition, right? For example, genes, certain kinds of genes could be disease-causing genes, and certain other kinds of genes could be genes that are risk factors for a particular disease, right? In this case, we say that certain genes again could be risk factors, right? And how can you identify that this risk is genetic in nature? See, suppose me and my brother at around 10 years of age, we both had heart conditions, right? So at 10 years of age, not generally people do not have heart conditions, but if we did have such a condition, it would indicate that maybe there is some genetic component that is involved, right? Because that would explain a lot. Similarly, congenital abnormalities or developmental delays in children are also usually indicative of a diagnosis of a genetic predisposition or a genetic cause of the disease, right? Also, there was another set of factors, which is uses of genetic testing at clinical level. So what kind of genetic testing can you do if you suspect that a disease has a genetic cause? What kind of genetic testing is generally done in clinics at this particular point of time, right? So some of these are newborn screening. So what is basically newborn screening? It basically means that, for example, a baby who has just been born and say that a clinician has observed that, okay, this child has a lesser circumference of the head as compared to the other children of this particular child's age, or this particular child has a lower body weight, right? So he might suspect a genetic cause for it. So that is when he might suggest the parents to go for a newborn screening. So what the screening will basically test for a lot of deficiencies, a lot of enzymes that are normally present in the body, right? It will be a genome sequencing, and it will test for a few hundreds of enzymes and proteins and bodily and such other bodily functions, and it could basically indicate that is there a genetic component that is involved in this case? Is it a lack of certain kind of enzyme that this kind of a condition is basically troubling the child or anything like that, right? The second kind is a carrier testing. So what is a carrier testing? For example, there was a couple, right? So, for example, there is a couple. The man has a brother, maybe, who is colorblind, right? And this female also has a first cousin who is colorblind, right? So we want, like, color blindness is not a lethal condition, right? I am just giving you an example. It could have been a lethal condition that we might have been talking about. I believe somebody is trying to join, so let me just admit them. Yeah, so okay, I'll share the screen again. Yeah, so see that some cousin, some brother and sister of both the people involved in this particular couple, the male and the female, have color blindness or any other lethal disorder, if you can think of that. Say cystic fibrosis, right? And this particular couple is very like afraid of whether their progeny could carry this particular disease, which is cystic fibrosis, which could be fatal or it could have adverse effects also, right? So what they want to do is that before they try to conceive, what they want to check is whether they are carriers of cystic fibrosis, right? So they will go and each of them will get their gene genome sequenced, and they can find out whether they are carriers for this particular kind of disease. For example, there is a recessive disorders, and if both of them are carriers, then their children will have a very good chance of actually developing that disorder. But for example, there are two different kinds of genes that can form that can contribute to a disorder. For example, I am delving a little deep into it, but I hope you understand. For example, in carrier testing, let me pick another example. For example, we say that there is a male who is deaf, right? This person cannot hear. And there is a female who is also deaf, right? I'm just denoting it as D. So they are basically impaired of hearing. And now they want to conceive, but they want to test that with how much possibility there is that if they conceive, their child, whatever child it is, boy or a girl, will have this possibility of being deaf, right? So deafness is, for example, a disorder that could be because of different kinds of genes, right? For example, if the problem or the defect in both these individuals lies on the same allele, then there might be almost entire chances that if they conceive, whatever child they have will also be deaf. But it might be possible that since it is a recessive condition and both of them have have different kinds of alleles that are involved in this deafness, what will happen is that they might be given a clearance that, okay, even if you conceive, we are definitely sure that your offspring will not be deaf. Why? Because you are deaf because you have a mutation in this particular gene, and your husband is deaf because he has a mutation in this particular gene. But since it is a recessive condition and only one of your genes will be involved and one of his genes will be involved, your offspring will completely be normal, right? So that is why carrier testing is done. I hope it is clear. The
Madam,
next
carrier
one,
testing can be done before marriage or after marriage.
that is completely up to the couple actually. So it is before conception. It has nothing to do with marriage over here, right?
But I think
So
there
it
is
is
one
up to
example. Cyprus country is there, which has brought out this because of this carrier testing, brought out percentage of thalassemia from 149 per thousand to 1 per thousand. That's what
the
that's why I was interested in this genomic thing. Because of
yeah.
just
Actually, it
counseling,
is
they had
very
and they
hmm.
had given result not you are carrier or you are not carrier. They had given the result just in the form of compatible. Your marriage is compatible or your marriage is not
Yeah,
compatible. Means they are not
yeah.
even revealing who is the carrier and who is normal. So in that
Yeah.
way, so means
Yeah,
the
so this is what
respect,
at a
authenticity.
3D. Yeah, so
Cyprus country,
your perspective, whoever is doing the study should know this. But what the other person needs to know is whether they should conceive or they shouldn't conceive. They are not really always like this explanation will not always be required by them. They will require an answer. So it might have been presented in that way, right?
that
So
is a
if we
small
come,
country. Before going to the church, they made it compulsory testing for thalassemia, which is not
for
feasible in India.
thalassemia, actually
Everybody
there
want
are
to
some
know what I am suffering from.
populations. There are some populations wherein these recessive conditions they flourish a lot. Why they do flourish is also one major problem is also consanguinity. Consanguinity is basically marriage between close relatives, right, or cousins. So there are some parts of the world wherein consanguinity is very common, and that is why what happens is that the chances of recessive diseases to basically show up in the population is more when it is a consanguineous marriage, right? So like these countries are one of those countries, right? So over there, it is actually a very good initiative that has been taken. And if we have our population is on the boost, we should have appropriate number of resources also to actually perform this, right? So even if we do have equipments, it is still a costly technique. Sequencing is a costly technique, so we need a lot of monetary resources as well to actually be able to perform this. Moving on to
Carrier
prenatal diagnostic testing. Yeah.
Madam, continuity, I am not against anybody, but I think one community, Parsi, is on the verge of extinction because of that hemophilia. Because males they are sufferer dying at a young age, only females still they are doing that consanguinity means Parsis, and in the families
Yeah,
also they are getting married.
yeah. So actually, we cannot really say that regarding one particular community or one particular country per se, because there are, for example, it is not just about a country or a religion or a community. For example, if you say that there is a new island, say 100 years back, there was a new island that was discovered, right? And in that island, maybe 10 people went and they actually started living on that island first. And now that island has around 500 inhabitants. And all those 500 inhabitants have initially come from those 10 people, right? So there is a lot of redundancy in that genetic pool, right? So this redundancy that comes with such a thing also for it is a normal genetic process also. So that is actually how these recessive mutations are actually surviving. But people are becoming more and more aware about it, and that is all we can do about it, right? So coming on to the next kind of testing, which is prenatal diagnostic testing. So prenatal diagnostic testing is basically once a female is pregnant, what they can do is what a couple can do or what the female can do is that they, for example, suspect that something could go wrong with that particular conception, right? For example, again, maybe later they realized that okay, we have a history of uh, maybe trisomy in our family, right? So Down syndrome is something that has occurred in maybe distant cousins of ours, and later it came to their knowledge, right? But now they are a little unsure of whether or not to continue with a particular pregnancy. So what they want to do is that they want to test whether this unborn child has that particular disorder, for example, Down syndrome or not, right? So what they can do is that at the earlier stages of pregnancy, when abortion is still an option, only then it makes sense to perform this particular type for testing. What they can do is that from the amniotic sac, they can collect a little amniotic fluid and they can perform genetic screening on that. And they can basically decipher whether or not the child that is growing inside is safe or safe from Down syndrome, or whether the karyotype has come out to become to be Down syndrome positive, right? And then it is up to the couple whether or not they want to actually continue the pregnancy. So that is one such other clinical testing that is performed, right? Another form is predictive or
Mainly, madam,
predispositional.
madam, sorry to interrupt. Mainly, it is chorionic biopsy and it is done in elderly primate to avoid Down syndrome. Because this PCPNDT Act is very stringent. So even if you
Yeah.
diagnose, so it takes too much time to terminate the pregnancy. So it's better now they are
Yes.
going for chorionic biopsy, and especially they in elderly primate who is chances of Down syndrome is more in them.
Yes. So, yeah. So this is actually how it started. But yes, now we have a lot of progression towards every individual testing also, and yeah. So that is actually one of the reasons why it is always advised to perform it as quickly as possible, because at that time, even if you take a decision regarding a pregnancy, it is more like doable. Like whatever decision you want to go with, it is like possible to actually go through with it rather than more later stages. But yes, there have been advancements in all of these screening tests as well. Also, prenatal diagnostic testing is one of the thing have not been updated in the recent years if it has been legalized as yet, but it was something that was not legalized in India. Why? Because, and some other countries as well. Why? Because people over here, they like used to take or there was a speculation, I think, that it could be taken unfair advantage of for basically not going through with pregnancies wherein female children were a possibility, right? So that is why a prenatal diagnostic testing does not really happen in all countries. But in the countries that it has been legalized, it has proven effective also. So, coming on to the next one, which is predictive or predispositional genetic testing. So this is basically just, for example, I were to go today and get my genome testing, and I wanted to know whether, like, what is the situation of the genes that I have, the kind of alleles that I have. Is it more likely that I will develop a heart condition? Is it more likely that I have this particular allele that will like which makes more cholesterol get deposited into my arteries? Is that an option? So these kind of things. Or do I have a particular gene which is more susceptible, by which I'm more susceptible to get cancer? These things could basically come out of predictive or predispositional genetic testing, right? And the final is that genetic tests may be used to confirm a diagnosis in a symptomatic individual or used to monitor prognosis of a disease or response to a treatment. So again, if somebody has some symptoms and we think it might be maybe due to the loss of this particular gene. So we test that, yeah, is this particular gene missing? And from that, we can maybe confirm our diagnosis. Or, for example, in case of cancer, we are treating a particular individual and we need to check whether that treatment is helping in suppressing the tumor. So we check for the tumor microenvironment, whether those genes, the tumor genes, whether they have gone down or not. So it helps to monitor prognosis for disease or response to treatment as well, right? Then there are certain types of genetic testings that can happen. The first one is cytogenetic testing, which is more like karyotyping, right? Over here, like some sample will be taken and chromosomes will be studied. For example, in this particular case, I am not sure if you can see it, and I'm not I can zoom it. So yeah, in this particular case, I think you can see now that there are two arrows. One is on chromosome 22 and the other is on chromosome 9. And chromosome 9 looks a little, this chromosome looks a little larger than its homologous chromosome. And in this as well, this looks
Philadelphia
smaller
chromosome.
than its, yeah?
Philadelphia chromosome in ALL.
Yeah, so it is one of the classic examples, which is Philadelphia chromosome, wherein some part of chromosome 22 has been translocated to chromosome 9, right? And this will basically put an individual to the predisposition of a particular type of cancer, such as leukemia or myeloid cancer, right? So they can, and this particular translocation is known as Philadelphia chromosome, just translocation between chromosome 9 and 22 that is already very well studied. It is known as Philadelphia chromosome. Then biochemical testing could be an option. Biochemical testing, we would do tests for enzymes in our body, for hormones, these kind of testing, okay? Wherein blood samples are taken, and from blood samples we analyze other biochemical, we perform other biochemical assays, right? Then molecular testing is another option. In molecular testing, what we do, we actually go and look at the protein level changes, the gene level changes. We could also look at the epigenomics, lipidomics, metabolomics, these kind of studies. It is a part of molecular testing, right? Also, for example, if we take cancer as an example of a genetic disorder, and we say that genetic disorders or genetic studies or functional genomic studies can be used for diagnosing subtypes of a disease, right? So, for example, we are considering a tumor, right? So we could use different kinds of functional genomics, such as epigenomics or genomics themselves, proteomics or single cellular RNA sequencing. And what it will do is that it will help to basically explain about what kind of changes have occurred in the genome of that particular patient. For example, RNA, single cellular RNA can basically tell that what types of subpopulations of cells are actually present in the tumor microenvironment, right? So then we can actually target those particular populations in that particular patient from which this biopsy sample has been taken. So it can help create both a database for further study and personalized therapy for a particular patient, right? For example, I will take another case of cancer, and I will say that 5FU is one of the therapies, chemotherapies that is used in case of a lot of subtypes of cancers, right? Not even subtypes, a lot of types of cancers. This particular chemotherapy is used, which is 5FU. Now, what this 5FU chemotherapy does is that to some individuals, it is very beneficial, but to some other individuals, it is very, very toxic and their health deteriorates very fast, right? So what scientists or researchers came up with is that they found why this 5FU is very, very toxic to some individuals. And they found that there is an enzyme, which is DPY. If this enzyme is deficient in a particular individual, or if it is mutated in a particular individual, then that individual is very, very susceptible to 5FU, and it will have a toxic response in that individual. But if it is not suppressed in an individual, this therapy can be effective for that individual. So over here, what these researchers have done is that they have basically performed normal genome sequencing, and they have found that there was no DPYD mutation, right? Since there was no mutation in both the normal genome that is present elsewhere in the body as well as a tumor genome, right? So this therapy, if we actually give it, it may be effective in this particular individual. But if we see that in the normal genome, this DPYD mutation is present, we cannot give this therapy to this individual because it will probably kill this individual, right? Or really deteriorate his or her health. In this third case, we say that in the normal genome, in the rest of the body, this DPYD mutation is not there. But in the tumor genome, this DPYD mutation is there, right? All the more reason to actually give 5-FU as a chemotherapy drug, because it will not affect the rest of the body, but it will severely deteriorate the cancer, right? So it is just another application of what genetic screening could actually, how it could actually help us, right? I actually have a lot of slides. If you have certain questions, maybe you can ask them. Because anyway, I'll not be able to complete all my slides today. So if you have any questions until now, maybe I'll solve them first. And
Please,
if you
please
want,
share. Madam, please share all the slides you have prepared so well. It will be a pleasure to have them. Thank you.
is that these slides that I will not be able to go through today, what I'll do is that I will include them in my next lecture, right? So we can begin with those slides next time, and maybe we can get done with the question part a little faster. We can include this in the next time slides, and we can continue with this discussion first, and then the next week's discussion, if that is okay with everybody. Okay, so any questions in today's class? I know that Ajay had a question that I was not able to satisfy him about. Anyway, I will suggest some reading material to you regarding that. And I will also help if I can, like I will try again if I can basically clarify that out for you. Apart from that,
Is an enzyme or a gene? What you had just said in treatment of
yeah,
cancer, that 5-fluorouracil.
Yeah, so it is actually a gene that we are talking about over here. It makes an enzyme, which is also named as DPY only. So, for example, Huntington is a gene which makes a protein which is known as Huntington. Okay. So
I think
sometimes
Huntington
the
was
genetic
that answer to your question. You were talking about positional cloning. Huntington group had described that positional cloning first.
Okay, I'm not aware of this.
Along with that, Francis Collins and Huntington groups had described that cystic fibrosis positional cloning.
Okay, okay, I was not aware of that fact.
You said that you want to remember something, while positional cloning is cystic fibrosis.
No, that was something else. I still haven't recalled that. Yeah, it was some like I was trying to remember what kind of mutation that was. So that I have not able to recall until now. So anyway, yeah.
I'm clear about intron, madam, intron and exon.
Yeah,
Because
so I would actually suggest you to go back and either read this splicing part from one of the books. Maybe if you can find a copy of Human Molecular Genetics, maybe you can look up into that. Or even I think Watson has this alternative splicing in it. I don't remember I have it, but or I will maybe share it along. When I will share my slides, right? I will add attach maybe 1 or 2 links. When you get that material, you can actually try to read a little about splicing. I will try to simplify it further if I can, and get back to it in the next class, that how basically I want you to understand it, right?
Madam, what my logic or my thinking is going on, just I want to elaborate it more. There are 23,000 genes
Yeah.
which are coding more than known 1 lakh proteins. It may be more than 1 lakh, means still we know just 1 lakh proteins. It may be more than 1 lakh proteins. Okay. Then the splicing occur at particular sites AG and GT.
Yeah.
Means the part which is cut between AG-GT is the exon and remaining part is intron. But it's not so simple ki the one AG-GT, it might take 3 or 4 sequences of AG-GT to cut that exon part. Means not single AG-GT is not responsible. Means either you have to have continuous run of 2 or 3 AG-GT before exon and after
No,
exon.
actually, that is not the case over here. Matlab, there are other possible mechanisms of splicing also that could occur, but this is the most common one that has been found. So I just picked up this one. But in this case, it is just GUAG only. It is not a multiple repeat of them.
And most logical for convenience, we are giving them name as an intron and exon. Means there is non-coding introns or means there are introns and exons, and there is another class of non-coding introns. We call them junk genes or like that. They are not non-coding exons. They are introns. So means part of intron that is coding.
Yeah. The non-coding terminology itself has been under a lot of debate recently because there are a lot of regulatory effects that have been found in
introns or exons, or call them non-coding. Means if there is non-coding, there must be coding introns. Okay, and as soon as you say functional genomics, means the counterpart structural genomics has to be there.
So okay, I would just to provoke your thinking. Where do you think tRNAs and rRNAs, these particular RNAs come from? They are not proteins, they are RNAs. Where do they come from?
No. They are not in the cytoplasm. They are not coming from the nucleus. They are part of the nucleus, but they are coming from nucleus to. They are from nucleoporins. They are diverting towards into the cytoplasm.
But how are they coming into existence? Like, for example, we know that we just have chromosomes. Let's say we just have chromosomes. Chromosomes are responsible for production of RNAs and for production of proteins, right? In a general sense.
rRNA, they are coming from nucleopores of nucleus, and then they are transferred to RNA. It's not just that cytoplasm is generating RNA. Means mother cytoplasm only contains mitochondrial DNA, not all RNA. So embryologically also
is DNA also contributing to some forms of RNAs. For example, if we talk about siRNAs or tRNAs, is DNA only making these RNAs?
DNA is only making these RNA, siRNA, and these all RNAs, they are made by DNA only. Na? It is coming from the nucleus and nucleopores. They are the guiding sequences. How much amount of RNA you require, that is RAS controlled, means just like a pedal and accelerator thing. It is a RAS controlled thing. How much amount of RNA is required in the cytoplasm for amino acids. So that's why if terminology of intron and exon is clear, then we can say it is the part of alternative splicing or not. If you ask any artificial intelligence software, is intron a part of alternative splicing? So they will answer yes. Intron can be a part of alternative splicing, but they are called as exons.
But introns are a part of alternative splicing. Introns are the regions that are being spliced out. What you need to ask is, are introns a part of mature mRNA?
But they are not. Before splicing, we cannot pre-decide if this is intron or exon. That's my question is.
Okay. According to me, actually, according to this terminology that is being debated a lot.
We are not researchers or Nobel Prize scientists. Let's see what is in the book. That's just my query is there.
Yeah.
You are researcher, you are scientist, You may get Nobel Prize,
Yes.
wish you get it earlier, but it,
Mhm.
it should be from the book and it should be authenticated. Where it is written,
Yeah, yeah.
the part which is retaining inside the nucleus is intron, and which is coming out of the nucleus is exon.
Okay.
Thank you, madam.
Okay.
Thank you for your
So,
valuable time and
this
allow me to speak.
That is most welcome for that. And yes, so on this note, I will also maybe discuss it with more of my colleagues, right? And take in what inputs or perspectives they have, and maybe we can discuss this again in the next class, right? So I will try to find out some reasoning or logic, better logic for this also. All right. So with that said, I can first, I forgot to stop the recording. I can stop the recording.